On this week's GARNet Research Roundup, I'm really delighted to be joined by Professor Andrew Fleming from the University of Sheffield. For the second time, actually, Andrew joined us last year to chat about a paper which involved Arabidopsis and moss stomata. In this roundup, we're just talking about a paper just published in Current Biology, the title of which is Stomatal Opening Involves Polar, Not Radial Stiffening of Guard Cells. And this paper is really interesting in that it really changes a paradigm of understanding about how guard cells seem to open and offers some new insight on that. So thanks very much, Andrew, again for joining us. And it would be great if you could give us an overview of this paper and what you found so interesting.
So, as you said, the paper, again, is focused on stomata. And stomata, as most of you know, are made up of two guard cells. And these guard cells swell and deflate, and that creates a pore between the two cells. Mm-hmm. This has been known for a long time, and if you look at stomata under the microscope, it's very clear that the inner side of the guard cells around the pore mm-hmm. are thicker than other cell walls. Okay. And this was observed over 100 years ago, and it became very quickly part of the literature that this inner thickening of the guard cells was really important for how guard cells work. Mm-hmm. There were some questions about it in the 1970s based purely on theoretical grounds. But the thing that we did that is probably different is that we started to use a technique called atomic force microscopy, AFM, which is a nano indentation process that allows you to go along and actually prod the surface of a plant cell or animal cell at a very small scale, and to actually measure the stiffness or the extensibility of the tissue that you're prodding. And what we found was that in mature guard cells, yes, absolutely, the inner side of these guard cells was stiffer than other cell walls, but in young stomata, there was no apparent increase of stiffening of the inner side, and yet the stomata worked perfectly fine. They can open and close. Okay. So the first observation was that this inner stiffening is clearly not absolutely required for the guard cells to work. And then using the same technique, we saw something rather surprising, or very surprising, was that there was a region of the stomata that were stiffer, but these were focused right at the tips of the guard cells, so not on the inner side of the guard cells. And that was quite a surprising observation, and therefore we followed up in the paper.
So you said that was a surprising observation, but did you have a clue? Is there a difference in morphology, or is there is it a thicker cell wall in the corners? Do you have any clue at all?
Nothing particularly obvious. I mean, obviously, it's an area where, like, actually three cells come together. Therefore, it is a bit thicker than the normal cell wall. But there are lots of other cell-cell junctions where this happens. So there's nothing overtly strange or outstanding about that region of where the two guard cells join, at least under standard microscopy techniques.
Okay. So what the nice thing is about this paper, one of the nice things about the paper, is that you link this cell biology and the AFM work with some modelling work as well. So can you give us an idea how you link those two things together to come up with your new hypothesis?
Yeah, again, it was somewhat fortuitous because we had started this project very much from the experimental side, from establishing the AFM technique. Mm-hmm. And then I think we just heard by word of mouth that down at the John Innes Centre, a group there led by Richard Morris, had been, again, for a number of years developing what's called a finite element model of guard cells. Mm-hmm. And so this seemed a good time to get together and to have a chat. Mm-hmm. And what they could do was to take our experimental observations and, if you like, test them against the model that they had created. Mm-hmm. And when you have experimental data and theoretical modelling data, and they don't quite jive, don't quite come together, then you can ask the modelers to explore their model to find out what is it they could change in their model to make it simulate a bit closer to the reality that we were seeing under the microscope. And that worked out really well. It's a really nice collaboration.
Cool. Well, it pays that. It obviously pays to chat and keep your eye on the literature.
It pays to chat.
So actually, looking at the model, so you've got some images of the model and the stress and strains which are put on the guard cells within the paper. So to me, when I look at it, it looks as though with the fixed point, when these fixed points at the poles, that you might get a slightly different shape of the stomatal aperture. So the hole looks like different. So were you able to measure whether there's a different shape, or is that something for the future?
It's something we haven't actually measured, but it must be an outcome of what we're seeing. Yeah. So no, it's not something we've actually measured, but it is something that could be measured in the future to back up what we're seeing.
Yeah,
although the shape changes might be quite subtle, I have to say.
So you said it looks like there's this stiffening in the polar regions of the guard cell wall. So what data have you got to suggest there's any differences in the composition of the cell wall in that place, in those locations?
So again, another collaboration that we've got going is with the lab of Paul Knox, who's just up the road in Leeds. Mm-hmm. And Paul's developed a whole panoply of antibodies against particular cell wall epitopes. So you can use these antibodies to look at your stomata and your guard cells and see whether there's any difference in distribution of cell wall components around those cells. And so again, using those antibodies and also a probe from William Willets up in Newcastle, mm-hmm. we could see that there's something special about the pectin in the region where these stiffenings occurs. Mm-hmm. Now, exactly what the precise molecular nature of that pectin is still, I think, open to discussion. Mm-hmm. Pectins are very complex molecules, but our data certainly suggests that there is something different about the nature of the pectin gel in the cell wall at the points where we're seeing the stiffening using the atomic force microscopy.
Okay, excellent. So the majority of this work is done in Arabidopsis, but you did do a little bit of work in both tomato and in corn as well. So can you say something about what you found in those other species as well, and maybe what it means for the future of agriculture even as well, potentially?
Yeah. So I mean, basically, we did we found it first in Arabidopsis, then we started to look at other plants that were around, and also that one could use the AFM on. The AFM is a great technique, but you have to have quite a smooth surface for the probe to work, mm-hmm. and lots of plant cells and guard cells are actually quite bumpy, and the technique doesn't work so well. But luckily for us, it works in tomato and maize, and we see essentially the same pattern. So there's this stiffening of the poles. Yeah, to say it's universal from looking at three plants maybe too much to say, but it's not just Arabidopsis. Okay. And clearly the main interest there is that if you look at things like corn, wheat, rice, the monocots, the grasses, they have quite a distinct guard cell stomatal complex structure compared to Arabidopsis. So in addition to having the guard cells, they have subsidiary cells around those guard cells. And so the next step in the project that we're pursuing is to, again, do some modelling work, because it could be that the geometry of the guard cells and supporting cells in grasses is important for their function. At least, again, that's what the textbooks tell us. Mm-hmm. And then also to see whether there's anything special about the cell walls of the grass cell walls, again, particularly in the ends of this stomata, that enable those stomata to function better than the typical dicot one.
So that sounds really good. So yeah, thanks very much, Andrew, for giving the overview of the paper, and definitely a paradigm shift in obviously quite a small area, but could be significant for the future of agriculture potentially. Who knows?
Thank you very much.